right, so our next speaker is going to be Caleb Harper, world-renowned, I should say. I actually was, during my vacation in Sweden, in Gothenburg, Sweden, I randomly ran into someone that had worked with him. So it's, on one hand, a small world, but then says something about MIT's reach. So he's the principal investigator and director of the Open Ag program at the Media Lab, and you got to wonder what the Media Lab knows about agriculture. So he's going to talk a little bit about that. He has a focus on high-performance urban farming systems, indoor farming. He has a background in architecture, actually, has a master's in architecture from MIT, and he's working on developing an open source platform for the hardware, the software, and the data that pertains to indoor farming. And he's also an entrepreneur. He has 2 startups. So with that, let's welcome Caleb.
Thanks. All right. So I'll skip a lot of background to get into the meat, but I know I'm around for panels after this, and we have Q&A for the last 5 minutes. And I'm going to move pretty quickly, and I hope that's OK with you guys. I lead a group in the Media Lab. They span electrical, mechanical engineers, computer scientists, data scientists, plant physiologists, phytochemists, dietitians, and an architect. And we work on this process that I'll call record, decode, recode, and I'll explain what that means. When you look at the future of food, everybody says food crisis, but we're all talking about a myriad of crises, whether it's not enough food, too much food, obesity, water runoff causing pollution, all of this kind of cacophony of food crisis. For my work, I tried to find 2 derivatives that I could work on that would underlay a lot of these kind of macro level problems to make it understandable. One of my sponsors for my research is a retailer in the United States. So I did a study of their produce with them, and then average produce across the United States. And one thing that we found illustrates the point. How old do you think an average apple is from the date it was picked until the date that you put it in your mouth in a grocery store in the United States? 6 months, 7 months. Any optimists? 9 months. Think about it when it's at your house and it rots in like a week. 14 months. So this is astonishing to most people. It is an amazing feat of engineering, the fact that we can hold apples that long aesthetically. But what we found as part of our analysis was that all of the antioxidants in that apple had been dissipated by the time it reached the consumer. So this is a meta point that underlays a lot of those issues, which is we have optimized for yield. We have not optimized for nutrition, and we have not optimized for flavor. And that will be the next great challenge of agriculture. The next one that I address in different ways, and Secretary Vilsack shared this with me before he left office, and I think he still may regret it. This is a study of the American population, what they would support or oppose in government policy, according to the last administration. A lot of things on here that people argue about and are highly political, but the one that's circled says mandatory labels on food containing DNA. I'd say 20% of the audience laughed. So 80% of the American public that was surveyed in this study would support mandatorily labeling DNA. So this tells you that the average person, being so far removed from everything we're talking about in science, doesn't really understand there's DNA in their food, let alone can they have a coherent conversation on GMO, the advancements of GMO, and how we've been doing that for 10,000 years. So a lot of education and transparency. So these are the 2 big things, a shift in what we optimize for and a whole lot more transparency. And you'll see every single company is betting on that right now. So we're in a moment right now that I think I need to call out before I even say what I do, which I would call bio-digital convergence. You know, the Media Lab, as was mentioned earlier, is not a home for agriculture, has not traditionally been a home for biology, has been a home for everything but. It was called the Digital Media Lab before it was Media Lab. So why am I working on this right now? And that's because this moment is when we've now gained and are gaining every day the ability to digitally control biology, program it, design it, and make it live. One of my colleagues, you may be familiar with CRISPR, and I'm not going to get into these. We can talk about them later, but you may have heard of CRISPR, and you may know what it is. But then there's already 2 generations past CRISPR. So while we're all busy talking about CRISPR and its litigation with some school across the ocean, or on the other side of the ocean, there's gene drive, which takes an edit, forces it into an offspring, which means that if you edit an organism, you can force it into an offspring, which means you can now ecosystem engineer. This is possible today. So the moral hazard of that has also pushed Kevin Esfelt in the Media Lab to start working on daisy chaining, which is an edit that's inheritable, and now gets spread across chromosomes so that when the animal, in this case a mosquito, or he's working on Lyme disease with rats, when the animal breeds, the edit gets weaker and weaker and weaker until it's mathematically calculable how to get it out of a population. I know that's a lot of information, and just wait, it's going to get worse. But this is to tell you that like the conversations that we're having now are already too late, and the engineering and the biology and the digitization of it are far, far beyond what we're talking about. I sit on the board of a nonprofit called New Harvest. New Harvest manufactures cell cultures from turkey cells, beef cells, fish cells. They support open source research in the space. Of course, there are a bunch of companies now rising up in that space that are making meat, that are making leather. If you aren't aware, just straight from a cell biopsy, feed the cell a feedstock, which in the case is mostly fetal bovine serum, apply it to some kind of structure, and then form a product on the other side. So we are moving towards cellular-based agriculture and cellular-based manufacturing of animal products. I would say, you know, it's still early, so it's not tomorrow, and everybody says, oh, the $500,000 hamburger. But, you know, this is the same way the genome went to, you know, whatever, $5,000 today. We're on a very clear path to where this becomes reality in the near future. As was mentioned, I have two startups that have spun out of my group. One of them is funded by Acre and Flagship, and one of the sister startups in that group is called Farmers Business Network. Farmers Business Network is an analytics service for farmers that was started by somebody from Kleiner Perkins and Cargill. And the idea is to give the farmers the hedging power in the market, because it's the farmers' data that's most important. It's really hard to get that farmers' data, so to get them to give them the data, they offer them a transparent marketplace, because inputs are basically the same cost, commercial commodity inputs, and they offer them that in order to get them into network. They now have more data digitized in field in the last 5 years than the USDA has digitized in its entire history. So our land is getting incredibly described. At the same time, another sister startup out of Flagship called SIBO, working on computational systems that tie their data with genetic information to create models and simulate how a plant would work in that field. Work on a project with Laszlo Barabasi over at Northeastern that's about pairing what I just talked about with our own human genome, and going from the food we eat, decomposing it into chemicals, taking those chemicals against our individual, as he calls, foodome, to prevent diseases that we are predisposed to have. This is all very early stage stuff, but happening all around. And of course, in the meantime, if you're following this one, Whole Foods, Amazon buys Whole Foods. The next day, Walmart kicks Amazon's vendors off of Amazon's cloud. The next day, Amazon releases a meal kit service, cuts Blue Apron stock price in half in a day. And then the next day, which was like just a few weeks ago, Walmart announces that all of its vendors over the next 2 years have to get on blockchain. So retail going crazy at the moment, changing faster than it has in a very long time. And this is because of all of these pressures from the consumer for transparency and trust. Now I'm going to talk about my work. Now that I've you zoned out because you're like, this guy talks too fast about complicated things. This is what I work on. First, I'm going to show you tools. Then I'm going to show you the data that we gather. And then I'm going to show you what we do with the data with some of our commercial sponsors in the lab. So first, tools. Generally speaking, if you know about the genome or genetics, I'm working on the phenome or the expressions of the gene. So inside of these boxes, and you've seen them in other places, but the way to conceive of them is we're designing a set of stress. Chemical stresses, atmospheric stresses, biological stresses. We are then applying them to specific genetics, and that causes a very specific expression. Another way of saying that is I'm building Tuscany in a box, I'm applying it to that Tuscan tomato that everybody loves to eat, and I'm growing the Tuscan tomato with the flavor that everyone says is only possible in Tuscany. So this idea is part of something that's really been hard to study, which is all the ways that the environment influences genetics to create nutrition and flavor. And it turns out we really care about nutrition and flavor and less about the genetics that we eat, because the genetics don't become a part of us and everything else does. So that's the meta of the tool. In the background, we collect as much data as we can. This is just an early UI, and you'll see some later ones. We collect as much data as we can, and we call it a recipe. So we're monitoring all the stress that's been applied to the plant. Then we're monitoring the expression of the plant to try to create correlation. One of the smallest, cheapest tools I've been working on for a while now is called Personal Food Computer. It started with me at the lab with 2 Dixie cups and an Arduino, has now, as you'll see, spread to 60 countries, about 2,500 people, and gone through a lot of iterations. This was all about a hacker-maker tool for doing what we did, because it's very expensive to work on the stuff that I'm working on from an overhead perspective. So I put it out into schools. I gave them a video game, this was super early too, where they had a digital environment, the sensors, information, and things they could change. They load a recipe that some other kid has grown anywhere else in the world. They load it digitally, and they load it physically. When they do that, they enact the last climate that was applied by whoever applied it. And then you can see on the left-hand side, as the plant is growing, that's about 3 weeks of growth, so it's not boring for the kids. They get to try things. They're like, CO2 is bad, so they crank down CO2, the plant dies, kid learns a lesson. More importantly, we download the data from all of those experiments, and we compile it all together. So this is how I'm building big enough data sets that can actually be useful for machine learning and artificial intelligence. One of our projects really shocked me. The kids, on their own with their teacher's guidance, used Farmer's Almanac data from 100 years ago in Massachusetts to rebuild the climate of the past. And you have to be a little creative on this one, because we all know it isn't the exact climate of the past. But they modeled the climate of the past. Then they used current weather data to model the climate of the present, and they used IBM Watson prediction to model the climate of the future. Imagine how much more interesting it is for a kid when they're trying to learn about climate science, and you're like, climate's changing, and they're like, no, it's not. Or they have the climate of the past, present, and future in their classroom, and they're growing inside of it to see what changes. Another scale of tool we call the food server, which is now manifested into these kind of shipping container formats. I can talk all about that and all the startups that are going for it. But I think the important thing to see for me is if I find something in the little one, that's cheap search space. Then I take that into the big one, and I scale it out to see if it's replicable. And then eventually we take that out into manufacturing. We've done a lot of different things in those environments. One is to play with how little water we can use and what the effect will be on the crop. This is an aeroponic system. Don't get too caught up in it. There's all kinds of ponics right now. People just make them up every single day. But all we do is we mist air into a root system. That gives it a lot of oxygen, nutrition, and it creates an incredibly healthy root mass. Think of it like giving the plant 2 hearts. So because it has so much more root, it can grow a lot faster. We've been able to do 3 to 5 times faster than field production. This was a trial with General Mills. This was a trial with Driscoll's on their strawberries. It was very early stuff where I was just piloting what does it taste like, how fast does it grow, because a lot of the claims in my space are not true. We've played with genetics. So this was to prove a point that we could bring back ancient and rare genetics. These were from seed bank. These tomatoes hadn't been grown in 150 years. They're supposed to contain more lycopene. Lycopene is good for brain health. So this was to prove the point that we have optimized for yield at all costs, but we have an entire genetic history of things that we have never grown for commercial production that may actually taste better or have better nutrition, and we're going to start exploring that. We did the same thing with beans and just about anything you can imagine. One of my newest projects, and I'll show a little bit about the sponsor with that if I have time, is the hazelnut computer. Same basic premise, a tree computer for trying to figure out how to reproduce the sun, how to reproduce day-night cycles, and how to see how the trees will do in advance of planting. Whew. You guys ready? That was just tools. This is data. So when everybody's like, big data, I actually like to show the data we gather and what we do with it so it makes sense. I call this the Open Phenome Project. We collect data across a number of different sets. One is atmospheric. This little sensor box was built by a 12-year-old off of our plans. It's temperature, humidity, CO2, all the atmosphere. We collect the information in the water using a spectrophotometer of all the individual elements that the plants are consuming. We take that plant forward into gas chromatography and mass spec. So if you don't know what that is, or these chemicals don't look familiar to you, they're secondary metabolites of the plant. This is what people call flavor or nutrition. So this is how we find out if you apply some stress, how do we quantify goodness on the other side of the plant? And I've gotten very accidentally familiar with analytical chemistry in the process. We also collect biologic data. So what you're looking at here are a bunch of Petri dishes that are samples from the water, so the root zone, the leaf tissue, and the surface. What you can see is there's a ton of stuff in the root zone. Not that much stuff makes it into the plant, and there's barely anything on the leaf surface. There is a startup in Cambridge that's funded hundreds of millions of dollars now called Indigo, which is also a flagship startup. that has just isolated 2 of those microbes, applied them to a seed coating, and then now are getting yields higher than wheat has seen for the last 10 years just by planting 2 microbes in the soil alongside the seed. This is incredible white space in science. It is just not known. So what we do is we sequence everything and we tag it with a time and a date stamp, just like all our other data as part of the recipe. The future of ag is images, but it is not the current. Images are cheap. We can get information from images. There's a lot of different ways to use images, from size and color differentials to anomaly detection to pest detection, but it takes a whole lot of images and to correlate to what actually is happening in the field. And so while I think drone, microsatellite, satellite is the future, right now is the time for trying to figure out what data pairs to what image to be able to do the learning algorithm to make it incredibly useful. This is a big struggle at the moment. So we produce high fidelity chemical data, high fidelity image data, and all the causal data so that we can say, if you see that plant, it has a high probability of having this chemical profile in the future. We use spectrometers, again, another form of non-invasive. This is a very good place to look into the future, where you're just getting spectral signature of the things that you scan. You're getting basically wavelengths and intensities that the eye cannot see, and you're getting close to a fingerprint with an image. So this is something I was working on with Target about them scanning in their supply chain so that they could say which tomato is actually better for you, which one is actually worse, and price accordingly. Again, images future, but building the correlations pretty tough right now. So these recipes, which is kind of what it looks like, pretty simple really. A state, the sensor reading, and a time, accumulates to about 3.5 million data points per recipe per grow. So as we grow anything, we're harnessing all this data. On my lab, I'm doing all the invasive, expensive things. Out in the field, they can't, right? It's just kids and makers, so they get images plus a little bit of causal. Then we put that into what we call our cyber-physical system, or our robot, because that robot is able to collect data and do things on its own through actuation. This is what our whole system looks like. We're also sponsored by Google to help us with our cloud. Cloud is very political these days, but our devices, unique hardware that's been built for sensing, tied in with a very somewhat crazy diagram, obviously colored in by a computer scientist, that represents now, every time I bring a bot online, they're infinitely scalable. With the power of the cloud and hooking up to things like BigQuery inside of Google, it's become very easy to do that. So that's the system. 3 minutes. Am I already past? Do I need this? Okay.
You got some time. You can take
Okay, because this is going to get weird. So we have all this data, right? 2 big buckets. What did we do? What did we get? Then we start using machine learning and regression analysis. So what you're looking at, if you've never looked at something like this, is kind of a heat map through regression. So the things that are darkest red are things that are directly correlated, like carbon dioxide and carbon dioxide. We know that's correlated. But what we start to look for is carbon dioxide versus potential hydrogen. In this experiment, we were analyzing wavelengths of light. And we start to see, are there correlations that we didn't realize existed? But this is pretty simple. So once you take that to a 3-dimensional search space, you start to see like kind of heat spots that recommend, in this case, that we use a certain fixture of light with a certain UV photo period, certain UV period with a certain photo period. What's cool about this is agriculture traditionally does all of these experiments to get here in 10 years. But if you can start training a model to do some of those experiments for you in search space, you can get to those answers very much faster. Of course, with any machine learning thing, it only is as good as the data that you put into it. In the beginning, since we had so little data and the machine had never tried to work with it, it literally predicted to turn off all the lights. If we turned off all the lights, all the plants would die. So that's the thing about machine learning, is if you don't have enough data and you're just learning, it's like a kid in a dark room that bumps into furniture and gets a bloody knee before they find the light switch. So this is how we're going forward. We published a paper on this recently. If you're interested, I forget the title of it, but you'll find it. It's a flavor learning paper. We take that model that we're starting to derive forward into simulation so that we can go and say, okay, if we were to change things, how would it change? So I mentioned the reprogrammable flavor. We also are now starting to work on this with nutrition. Just to give you a sampling, a single basil plant, 303 chemicals with 974 disease correlations. You don't think about it, but a basil could be designed as a pre-diabetic basil, or a basil that is good for heart disease, or certainly a tomato that's good for heart disease. So we have a collaboration with Northeastern and Harvard around, can we link the nurses study, which is the longest longitudinal health study for what nurses ate and what correlations it had to diabetes and heart disease, can we link that with chemicals, which is what Laszlo's group's working on, all the chemicals for the thing we ate, and then program food to respond accordingly. So that's kind of fun. This one's a little bit on the wild side, but we took our model. This is 30 years of climate data, the weather stations from the EPW, which exists already. We scraped that data, we put it into recipes. We stuck it through our model, and it created this map of the world. This is a fake map of the world where the best quality cotton would be grown. Then we correlated it against the FAO's data for the last 30 years of where cotton was grown. And we were about 75% correlated on our first shot. So this tells you that ability in the future to predict forward where we would plant for what expression is really nearing us. So I'll close really quickly. I do this. This project is about what can you grow in a box, and it's a cotton project with the world's largest cotton manufacturer in India. I do it with Ferrero on the hazelnuts for all different kinds of reasons that we can talk about, but that's more about where would they plant hazelnut orchards in the future, where would they acquire land now, and the last thing. We put all of this out open source because I'm trying to get the most amount of users on the biggest platform in the space to accumulate the most amount of data. I'm following in the footsteps of these guys for all the reasons that you know they've done open source, or at least pretend to. We put it all out on Wikipedia. We then created a community, a Reddit-style forum that gathered these people to ask and answer their own questions. It looks something like this. These are students, makers, teachers, hobbyists. I'm going to close really soon. This was with National Geographic. We throw make-your-own food computer camps. They come to the Media Lab, they take them home, then they start their own chapters. And they've called themselves now the Homebrew Food Computer Clubs. So if you remember, right, homebrew computer clubs, weirdos doing things that people didn't necessarily believe were going to be important. That's this generation. Those are the tools and the applications I've shown you throughout the presentation. Close with this slide. In 2 years' time, with no funding to anyone, an expensive project, and a hard-to-build machine, we've scaled to 62 countries and over 2,500 people on network. That's the end of the presentation. Thank you.
We'll do one or two questions. Any questions at this point? You're going to have to catch this, too, so this is maybe deterring people from nothing.
Even the simple questions.
Yeah.
Anything.
All right, so there's one over there.
Bueller.
Why did you start with tomatoes and basil?
White tomatoes.
Basil, quick turn crop, short growing cycle, doesn't take much energy, and can produce a lot of secondary metabolites that are associated to disease. They also happen to taste good, and one of my sponsors is a retailer, as I mentioned, so that's not a bad bump. That was why basil and tomato. Tomato because we just wanted to show that we could do it, and we had a few people interested in the lycopene levels in tomatoes.
So what was the result of that experiment that the kids did with the climate of the future, the present, and the past?
We're screwed.
Like, where did they?
Yeah.
what did they find?
Actually, it's kind of illustrative. Like, in the middle of the United States right now, they're starting to plant cotton. They never planted cotton before. So the North is actually in an advantageous position because we're going to be getting warmer. So it wasn't as bad as everyone would have expected.
Which cotton producer
Use
you're
the mic,
working
please.
with in India?
Wellspun?
Okay. Never mind.
All right, so pass that on.
You guys can just say, this is a small room, right?
Are you doing any work with food allergies?
Food allergies.
Like genetically tailored basil is great if I'm allergic to basil.
Yeah, no, I mean, I kind of stop at the line of trying to understand how plants think and how they produce the biochemicals, and then someone else will take it forward, hopefully, I think.
To get a group of middle and high school kids hooked into this, what's my first step?
Go to the wiki page, OpenAg Wiki. And actually, as of October 15th, we release our next model. It'll also go out in the Wall Street Journal at the same time. And it's much easier to build, much cheaper, has curriculum. We're in 25 schools in Boston for the STEM month, where they're building a lunar colony. And so if they're middle schoolers and they happen to be in Boston, they might already be exposed over the next October. But other than that, the wiki and the forum.
All right, so let's thank Kayla. We got to move forward here.